I was looking back at the history of the disease that started in 1994 on DeGray Lake, Arkansas, where 29 bald eagles died over the fall and winter with no known cause. And the research team that was mounted to study it in the next year didn't document any disease, of course. And when they disbanded the following year, another 26 eagles died, and they also realized that the American coots at the same site were affected. Let's see, I think it doesn't want to go forward again. Oh, there we go. The birds on these sites, both the eagles and the coots, appeared to be neurologically impaired. So the eagles had trouble flying, had drooping wings when they were sitting on a perch and stumbling on land, much like these coots are. It's a little easier to make a living as a coot. You just got to kind of swim around and eat the hydrilla, but when they would come up from diving, they wouldn't be able to right themselves in the water. And when they were on land, they were getting, by the time they get as sick as these two were, they weren't even able to feed on the hydrilla very well. The only way to diagnose this disease still is based on the original definition of the disease, which is that there are open spaces or vacuoles in the myelin sheath of the brain, and hence the name vacuolar myelinopathy. These are actually similar to some of the diseases in humans, like MS lesions, create the same sort of open space in myelin sheath. But this was the first time it had ever been documented in eagles or waterfowl from an unknown cause. And all other diagnostic examinations were negative or inconsistent. They didn't find that there were other problems in the histological examinations, like the liver, kidney were pretty normal. They didn't find any consistent toxins in the bodies. So they were just coming up with a big mystery about what was causing it. Initially, when it happened, that was the first time that it had occurred in Arkansas, but once they started to investigate it, they realized there were actually other sites that had the same kind of scenario once they knew what to look for in terms of the coots acting very strange and having eagle mortality. And all the AVM sites were man-made reservoirs. They had pretty low nutrients, really, no harmful blooms, but they had these dense infestations of invasive aquatic plants. And primarily, it's Hydrilla verticillata, although in the first year of the disease at DeGray, they were dominated by Egeria densa. So two other species which are in these AVM lakes are the Eurasian water milfoil and Brazilian waterweed. But as you can see, all of these species kind of fill up the water column with biomass and present a lot of substrate for a cyanobacteria to grow on. And I did find a new species of cyanobacteria that was growing in all the sites where the birds were dying, growing on the hydrilla, and mysteriously just on the underside of the leaf. But even though these are microscopic colonies, when they're dense, you could see them as spots on the underside of the leaf. Since it was a new species of cyanobacteria, I got to name it, and so I named it Eagle Killer Living on Hydrilla, sort of dramatic. But if you were looking for it on hydrilla that you would collect in Florida, you would look for these spots on the underside of the leaf. I carry a hand lens or reading glasses to help me magnify it a little bit. But if it's dense, you can see it even in the field. But under the microscope, I look on the backside of the hydrilla leaf. Under regular light, it's pretty cryptic. You can't really see exactly where the colonies are unless you know what you're looking for. But once I turn on the fluorescent illumination, I can see the cyanobacteria pigments, and those show up really well against the background. And if I turn off the light and just have the fluorescent light on top, then we see only the cyanobacteria, and we don't see the hydrilla leaf at all. So cyanobacteria are sort of like algae, and they're sort of like bacteria. They do photosynthesize, but they also have some of the characteristics of a bacteria. And they are growing epiphytically on the hydrilla leaf. So it's a surface for them to grow on. So what we have here basically is harmful algae that's growing on invasive plants, which is unfortunately a good way for it to move up the food chain, because the coots are eating the neurotoxic hydrilla and getting sick, and they're easy prey for the birds of prey. And the vet school here at UGA did some experiments that verified that toxin transfer. We found a lot of other birds that are also affected by AVM. Some of these are birds that hunters would target, including the Canada geese and mallards and ringneck ducks and buffleheads, American widgeon. So we actually do have some concern about whether or not this will be an issue for waterfowl hunters that are just consuming the meat. We know there's toxin in the gut contents, but we just now have enough information about the toxin itself to be able to test in the meat tissue. So that's something we're going to be doing this winter down in Florida and also here in Georgia. As I said, this is a seasonal disease, so strangely, it is only really occurring from, say, November through January. So in Florida, the timing is slightly different, but it seems to be following that same time window. And we do see the food chain transfer from toxic hydrilla, which is toxic by way of having the toxic cyanobacteria growing on it, that's fed on by coots, and they get sick, and the eagles consuming them then also become impaired and generally die from that toxin. So we first validated this hypothesis by feeding mallards the hydrilla that did have the cyanobacteria on it, and then hydrilla from lakes with no bird disease that didn't have this new cyanobacteria growing on it. And we found that only those ducks that received the hydrilla plus the novel cyanobacteria ended up getting those characteristic AVM lesions, those open spaces in the brain. And we have further processed the hydrilla by freeze drying it, grinding it up, and extracting it in a variety of solvents to see which solvent it will come out in. So it does come out in the methanol fraction, which would indicate that it is somewhat water soluble. We have a little more information on that now, and we realize that there are actually two compounds. One is fairly water soluble, the other is more lipid soluble that the cyanobacteria makes. So, but the bottom line is that you can extract the hydrilla material with the cyanobacteria growing on it and have a toxin that you can use for characterization. Oops, let's see. And we also started using this zebrafish bioassay to test the material. So previously we used chicken feeding trials or mallard feeding trials to see if the material was toxic, to see if they would get brain lesions. But we didn't really want to keep having to kill the birds to be able to test material. And we also wanted to learn more about how that toxin was affecting a zebrafish, which is a real good assay for looking at problems with vertebrate animals. It's a model animal for that because they know so much about it, how long it takes to develop, and what changes occur when it's affected by toxin. So when, as I said, we will dry the material down, concentrate it, and then those little exposures can be done with multiple larvae in a very short time window. They're only 6 to 7 days old, and they unfortunately respond pretty quickly with both, we see mortality in the little zebrafish larvae, but we also see a neurologically impaired behavior. So those that have been exposed to the AVM toxin often are like lying on their backs, can't right themselves. They have this their little pectoral fins are just flipping all the time and not really moving them anywhere. The regular zebrafish, the control zebrafish, are kind of zipping around their little dishes happily, but there's a real difference in what these AVM exposed toxin zebrafish are acting. So we had a number of different ways of detecting the toxin from, as I said, that AVN bioassay to see if they develop lesions, the zebrafish bioassay to see if there's mortality or behavioral changes. We also had a cell line bioassay and this invertebrate assay. So all of these are helping us be able to detect the toxin in the material and refine it so that we can characterize the toxin and go on to analysis that way. And of course, Hydrilla continues to expand. While we're doing this research, initially in Florida, the dioecious strain, and then as the monoecious strain came in from the 70s, it's been moving on across the country. See if this will run for us. So from 1952, there are documented locations in Florida initially, probably from the aquarium trade. Someone dumped a tank out the back. And as early as 1970, there was documentation of the monoecious strain coming in to Virginia. The first AVM occurrences were not documented until 1994 in Arkansas. And initially there were 4 sites there, but fairly soon thereafter they discovered there were sites in Georgia, South Carolina, North Carolina, and Texas that also had birds with AVM lesions. And much more recently, we have documented it in Florida since 2009. And we now have 5 sites in Florida where we're tracking that because we have confirmed that the Autochthonous hydrilicola, the cyanobacteria, is present, and we have documented toxicity in those sites. Our very worst site in terms of eagle deaths is J. Strom Thurmond Reservoir, which is about an hour and a half from Athens. So we've spent quite a bit of time over there, and we've lost 106 bald eagles in that location. And most of them have been verified as being AVM positive, but the only way to say that a bird is AVM positive is to get the bird recovered when it's still very fresh and getting that brain into formalin so that it stays fixed and you can do the analysis. If it decomposes or if the eagle is frozen, then you can no longer determine whether or not they have lesions because there's too many artifacts from either decomposition or what the freezing does to the brain. So it's kind of complicated actually to document these birds of prey and being killed by it in the field unless you have agencies that are responding very quickly to it and can ship it to the veterinarians to do the pathological analysis. We have documented it also in a number of other taxa by some feeding trials that we have been doing here at the University of Georgia. We know that herbivorous fish and frogs can get it directly by consuming it, but then we've also seen some toxin transfer. Herbivorous turtles have been confirmed with it as well. And I'll just show you briefly some of the experiments that students here at UGA have done to confirm some of these other pathways. So of course, Chinese grass carp are a good control for hydrilla, and we were worried that if you used grass carp to control it in a situation where you have toxic hydrilla, that they might become a means of toxin transfer, or it might just kill them. And we have done enough trials in the field and lake settings that we know it's successful, but unfortunately we also know that the grass carp can get the lesions and that there is some mortality. So there's definitely some risk in using the grass carp. We think that it's much smaller if they're very large. The only time when we really had problems with a lot of mortality in the grass carp was very small ones that we used in the lab trials. So the 10 to 12 inch fish they use for typical stocking shouldn't be a problem, but we still have concerns about that toxin transfer. And the fish get the same kind of lesions that the birds do in the optic region of the brain, which is not surprising. They're sight feeders and using that a lot. We have shown that if you can eliminate the hydrilla from the areas where the birds are, that you will eliminate the disease. This is one of our sentinel mallard trials in a reservoir where we had a treated and untreated coves, and this poor mallard was in the cove that still had the hydrilla and the autochthonous hydrilla on it. And they seem at first to kind of have almost convulsions or seizures, but eventually they will become almost paralyzed. The turtles have some problems with impairment as well. When they were, we did some experimental feeding with them. They have a lot of weakness and lethargy. They tend to not eat by the end of the trial, and they have this awkward gait. They're not walking very well. They also get lesions in the optic region of the brain. We saw that tadpoles, when they consume the hydrilla directly, will experience mortality pretty rapidly, and they just swim in circles, like, or swim into a corner. They definitely show behavior effects very quickly. And we know that these are things that are living in the water at the time when the hydrilla would be toxic to them when they're consuming it. Another animal that would be at risk are these little mole salamanders that have a planktonic swimming stage called a paedomorphic stage. They will be consuming some tadpoles and snails, which also are moving toxin up the food chain. And we did a feeding trial in the lab that documented both behavioral impairment and some lesions in the cervical spinal cord and olfactory tract. So slightly different part of the brain, but typical, the same type of lesions. And also saw trophic transfers through fish to water snakes, and they also had lesions in the spinal cord, which of course would make sense for a snake. That's a very important movement area and olfactory area as well. So I just want to finish with some of the concerns in Florida very specifically. We're looking at the risk to the snail kite because it's consuming especially these invasive apple snails as they continue to kind of take over the native apple snails, and they can eat the invasive apple snails capable of eating an awful lot of hydrilla, and snail kites are actually getting pretty good at catching them and eating them, finding them and eating them rather. And so we have documented in the lab that these snails can be do toxin transfer, and we have documented coots that had toxin and lesions in the field. The worst place in Florida so far that we found is Lake Toho, and specifically this Goblets Bay region. It does seem like that these denser areas, this is also an area where we found a lot of toxicity associated with the vegetation and snails. And we've continued to find a little bit more each year. We've gone down. I don't know if that's expansion or getting better at looking for it, but I do feel like there's a concern that it is expanding. As I said, we did document that it can transfer through snails to those chickens in the lab. We haven't documented a snail kite with AVM in the field, but it is rather difficult to recover them in time to do the analysis. So we're going to continue to be watching out for any snail kites that might have any impairment. It seems like it's growing on other plants as well, especially the invasive Hygrophila and Limnophila in East Lake Toho. So other invasive plants can support the toxic cyanobacteria and might be a problem in locations where there's not as much hydrilla, but these plants are dominating. So I want to leave time for anyone that might have any questions and acknowledge the support from Florida Fish and Wildlife Conservation Commission and U.S. Fish and Wildlife and some other agencies we worked with all along. The Army Corps of Engineers was really instrumental in getting all of this started, and we've had some nice support from the Gulf and South Atlantic aquatic invasive species, and a whole host of graduate students who do all the work, well, a lot of work anyway. Thank you.
Great, thank you very much, Susan. I'll just start off. I have a question that I think you I'm sure you answered, but just for my clarification, the cyanobacteria, it's a naturally occurring cyanobacteria that is just taking advantage of the environment created by these invasive plants to multiply to a degree that it's impacting the wildlife?
Wow,
or is that something that was introduced
very
as well?
well stated. That was good. We don't know. I think it is absolutely a cyanobacteria that either was so rare in that environment until the invasive plants came along and provided all that great substrate to grow and become very abundant on, or it came with them and our environment is allowing them to be very abundant relative. But I really still want to go to China and Taiwan and India and some of these places that are the origin, perhaps, especially of the monoecious variety, to see if it's there. We don't really know it because it's a newly described species that had never been seen anywhere, and we did all the genetic characterization on it, that it wasn't in GenBank and it was not a known species, and this is a novel toxin that it's making. So yeah, we don't know yet where it came from. But it certainly was not a problem until Hydrilla became very abundant on these reservoirs.
Okay. Any other questions for Susan before we move into our SISMA update?
Susan, I wanted to ask, I'm sorry, what are the most direct control efforts that we've got going on in Florida? I'm way up in the Panhandle, so and our agency is large and very diverse, so a lot of the efforts down in Central and South Florida, sometimes I'm not aware of, especially with some of the listed species that we have in those habitats there.
So I think any control of Hydrilla specifically is going to help with controlling how much of the cyanobacteria is available for these birds for it to become a problem. And so far, I haven't found that cyanobacteria growing anywhere sort of at that Panhandle region. It does seem like, and I've sampled some of the springs that are kind of on the western edge of Florida and the northeastern area, and their conductivity is really high, and we have never seen the cyanobacteria be abundant in really high conductivity water, like lower alkalinity, lower conductivity water. It's so far, that's for whatever reason, that seems to be a barrier. For instance, Okeechobee, we still haven't found it in there, thank goodness, but it's considerably different in terms of alkalinity and conductivity than Istokpoga, where we have found it. And I can't explain exactly why, but it probably is important for me to continue to sample Hydrilla throughout Florida just to keep tabs on that. It's a fairly simple way to see whether or not there's any risk in that location.
So do you know if there are some pretty intense efforts or targeted efforts around the hot spots that you're seeing in Lake Toho to eradicate or remove or control?
Right. So FWC is I keep them apprised of any locations where we find the cyanobacteria growing more densely, and they do put a priority on doing hydrilla control in those locations. And I certainly think that's been effective to try to at least hit the hot spots. Obviously, we're not going to kill all the hydrilla in Florida, but I think by trying to target those locations we know to be a risk, it makes sense to put some extra effort there.
Okay, thank you. Emily, are you ready for me?
Oh, I'm sorry. I was muted. And can you hear me now?
Okay.
Okay. Yes, I'm ready. So you should be able to start clicking to the next slide. Give it a try. You may have to hit the button a few times. There you go.
There we go. Okay. All right. So I'll just start out by saying thanks for having me today. And I've been with Florida Fish and Wildlife now since January of 2017. I'm a native of Scenic County, so I have worked in the Panhandle region for about 20 years, and have been involved with Six Rivers SISMA since its inception back in 2009. Let's see if I can get this to move. So as you can see on the map, this is our region as it is currently. We are unique in that we have Alabama counties and partners also incorporated into our SISMA, and it ranges from west from Baldwin County, Alabama, all the way over to the eastern border, which is Holmes County. Now this map shows Washington, so I think we do need to update that map on our website, and I know that Chuck Bergeron is helping us to make some changes to our website. Okay, I'm trying to get it to move forward. There we go. Okay. So, alrighty. So in the last few years, I've been kind of in and out of the SISMA in different capacities, and we've had a couple of different folks that have led our SISMA since the Nature Conservancy stood up the group back in 2009. We originally were started by a grant. And so we had a pretty structured strategic plan that identified subcommittees that I've got here on the slide. We had those subcommittees established and a work plan established, but there was not any update of those of that strategic plan for quite a long time. And after the grant period ran out, I think the group just kind of transitioned into more of an informational basis. So currently, I started leading the group last year in the spring. Our steering committee consists of several different partners from IFAS, DOD, Mark Gibson with Pensacola NAS, Craig Iverson with Blackwater River State Forest, and Dexter Sowell through the Florida Natural Areas Inventory. So I really feel that one of our best assets is our steering committee, and we've had some really good conversations, even though we don't have many opportunities to meet physically. We do have a lot of email conversation and some phone calls that has helped us to try to refocus our efforts here in the last year. Our meeting held in August of 2017, we had 14 agencies and about 24 partners who showed up to that meeting, which was probably 3 times as many folks that attended that we've had in the last 2 years. So we were really happy to have that many people attend the meeting. We had a great technical presentation by one of Stephen Enlow's graduate students on treatment of tallow trees and other common invasive species trees. And we had, we were able to get sponsors for lunch and also had an EdMaps training session during the meeting. So I thought that went really well. There we go. So, so some of our short-term goals, we realized that we're not getting a lot of engagement from our partners, and that buy-in is something that we really need to work on. You know, we had identified subcommittees, and we had a lot of new people in the room when we all met last August. But once everybody left the room, we just really didn't have a lot of response to email. Nobody really shared their projects. I would send out an email asking for, workday ideas or volunteers, and just wouldn't really hear a lot from our partners. So we know that that's something that we need to work on. Our steering committee worked to condense our strategic plan from a really long, burdensome plan that was about 15 pages long to something a lot shorter, and we thought more simple, but after talking with several SISMAs and Sherry and Sam, we're still hearing that we need to really focus on the simple and think more out of the box. And so with our June 7th meeting, we've been able to get a facilitator from FWC who's going to be attending, and Sam's going to come over and present also about, the benefits of the SISMA. Hopefully we'll be able to identify some strengths and weaknesses that our partners feel are important in moving our SISMA forward, and we can even possibly take a harder look at our strategic plan and break that down. It may look completely different when we get to the end of the meeting, and that will be great. We just I'm really hoping to hear more from what our folks, want out of our SISMA. And so these are some of the things that we've done over the last 6 months. Oops, it went by itself. Let me try to go back one. Just 1 second. There we go. Okay, so during the Halloween timeframe, we were able to conduct quite a number of EdMAPS trainings, but still mostly over in the Escambia, Santa Rosa western side of the Panhandle area. We had 3 training events. We had pretty good participation from 3 counties, Okaloosa included. Most of it focused on Kogan grass. Kogan grass is one of our big bad boys over here. There's a lot of talk on Kogan grass, a lot of workshops, a lot of trainings. So we picked that particular invasive to focus on. You can see the numbers. We had pretty good reporting for that November data collection. And then our Gulf Coastal Plains Ecosystem Support Team is that go-to group for us. They assist with our control efforts on most of our work days, and they've been doing a lot of Chinese tallow control in Big Lagoon State Park, which is over near the Perdido watershed. So our animal targets have been primarily lionfish, and we have a big lionfish removal and awareness day every year. Last year, you can see over 6,000 lionfish were removed. And so what we've been working on recently, primarily through the NOAA Sea Grant and our extension office here in Escambia County, is to get some pre-collection numbers and also our this wholesale setup. And what that's about is that we've got, divers now can actually apply for saltwater products license, and from FWC, they can, in that process, they will get their application. The application gives you a list of the licensed wholesalers who've been approved to purchase lionfish from divers. So once the divers spear the fish, then they can sell to those approved wholesalers. And right now we have 15 licensed divers and we have 5 wholesale buyers. So we really hope to increase this idea and kind of spread this awareness in an effort to help out with lionfish removal. This next bullet on Cuban frogs was supposed to have been Cuban anoles, not Cuban frogs. although we do have some Cuban frogs, but most of the work being done on Cuban frogs is more of an education and awareness. But the Cuban anoles are now established, and we are logging reports in EDMAP for the brown anoles. We are trying to help some of the Amnesty Day folks with FWC to do a little bit better job at the Gulfarium. The numbers have been low last year, and so we're going to work with the Gulfarium this year to hopefully make that a little bit bigger, better event, and possibly look at even a second Amnesty Day or possibly a different location in 2019. In education opportunity in Santa Rosa County, you know, this one was brought to me by Santa Rosa County Public Works, where there's a big stormwater pond project, and they want to remove some red-eared sliders. It is adjacent to a big butterfly house. It's a really big educational opportunity for us, we think, and so we're going to try to use that removal of these red-eared sliders as a way to increase public awareness of invasive species in the county. And this was pictures of one of our work days that we did along Scenic Highway in Pensacola back in October of last year. This was probably one of our bigger work days. Again, I haven't gotten anything from the eastern part of the SISMA. We did remove 60 tallow trees from this park, and we've been requested to come back, but like always, you know, volunteers are always a problem, just trying to get enough folks to get back out there and do it again. From Baldwin County, they've been working with the city of Daphne to remove invasives over around Eastern Shore. And there's a list of the species that they've been targeting. Lots of our forestry service partners, again, are targeting our same old tallow trees and coconut grass. Just yesterday, we had a coconut grass partnership workshop up in Andalusia at the Solon Dixon Center. And what we're trying to do there is come up with a successful funding initiative to fund a full-time person who will take over the legwork of managing a coconut grass work group. We weren't accepted for one funding initiative last year, an NRCS application that we submitted, but we've got a lot of buy-in, and so we're still working on that. Covington County, another one of our Alabama partners, has money, and they are actually getting landowners to call in with their coconut grass infestations and having those treated. So that one is moving forward. Blackwater State Forest is working on cattle repair with some of their new park rangers. Escambia County in our SISMA doing some good work over there on a wetlands preserve on the west side of the county. Again, same sort of species. We're also, FWC specifically has been working with them a little bit, and I'll send a shout out to Sherry on that. She sent me a template, and we've passed that along to Escambia County to help them with improving some of their management efforts for their work as a whole in Escambia County for removing and managing invasives. And we were having a workshop tomorrow with the private landowner teaching methods and techniques, but we have some bad weather coming, so we did have to cancel that for tomorrow. We are joining ARSA, our neighboring SISMA, for an invasive day on June 6th. And then, of course, I've already mentioned our June 7th meeting that's upcoming for our SISMA, and we look forward to having some good things come out of that. And that's it.
Okay, great. Thank you so much, Crystal. That's a lot going on, so that's awesome, and really great to hear, you know, some kind of refocusing and getting some new people involved. Any questions or feedback for Crystal? Let's see. And I want to go back really quickly because I think I was muted at the end of Susan's presentation. And so thank you again, Susan, so much for your time. We did have one question come over the group chat from someone who's having some issues with their audio. So Ian just was wondering if you, Susan, if in your research you had identified a temperature barrier to the cyanobacteria. So are there certain temperatures you find more conducive or ones that they can't survive in? And you're muted, so if you can unmute your mic for us. Sorry.
Yes, so
There we go.
cyanobacteria are extremophiles, and generally they can handle wide-ranging environmental conditions. We have found it in really warm water, and we've also found it in water that was near freezing. But the interesting part is that we haven't seen toxin production by the cyanobacteria unless it's below 12 degrees Celsius, which Florida doesn't actually do very often. So it's been interesting that when y'all have a very harsh winter, those are the times when we have actually documented toxin production in Florida. So I think it will be less of a problem in general in Florida unless there are times when it's really cold. And that's just something that we have noted from the field. We are starting we have some cyanobacteria cultures, and we're starting some experiments with them to test for temperature initiation of toxicity. And we also have some information that bromine may be a cue to toxin production. So it may be a combination of some environmental chemistry and availability of ions, but also a temperature that might affect whether or not plants are senescing and releasing some of those compounds. So sorry, long answer, but temperature affects toxin production probably.
Okay, great. Thank you so much. And Ian, thanks both of our presenters via comment. And yes, I just, again, want to reiterate, thank you so much for your time and both of you putting those presentations together and sharing that information with us. At this point, looking at the model from previous calls, Rose would usually do a shout out to the work that's been done over the last, since the last call. And I am still catching up on kind of all the reporting and everything. So I know she's got some great tools in place, and I'm still just kind of catching up on those and reporting on those. So I just kind of wanted to give a shout out to everybody that's on the call, the work you're doing and the participation, and the partnerships and the communication is all critical to the success of FISP and the SISMAs. So just wanted to give a group shout out, pat on the back to everybody on this call, and thank you so much for everything that you're doing. Just a reminder about the SISMA calendar for those folks that are involved in SISMA. Keep an eye on it. You can find the information about the monthly calls, and then if you're a SISMA lead, definitely be posting your events there to share, and that can be found on the web. And then next month's call, May 23rd, go ahead and mark your calendars now. We're going to get an update on some of the invasive plant species and their biocontrols from Dr. Kuda, so that should be really good. It's always good to check in with him and his team and hear what they're doing. And then we'll have an update from Treasure Coast SISMA. Moving along, here's our schedule for June through October. So we've got a great lineup of presenters and SISMA updates. So I'll touch base with the presenters and the SISMA update folks to double check on those, but we're always open to suggestions. So, you know, if you hear a presentation somewhere or have a topic that you really want to hear a presentation on, you know, please reach out to me and send me your ideas. You know, we definitely want these calls to be useful in presenting new or updating information as needed. So, you know, please reach out to me if you have anything you think would work for these calls. That would be great. Okay, and then just again, another reminder, we do these monthly with the exception of November and December. Join the CISMA's listserv for announcements about the call and really just lots of other things to stay in touch with all the work we're doing across the state. At this point, I do want to just check in. Let's do a time check. So we've got about 5 minutes. Does anyone have anything to add? Any other updates they want to share? We have a few minutes for that. And again, just don't forget to unmute yourself like I always do if you do want to chime in.
Hey, Emily, it's Sherry.
Hey, Sherry.
Hey, I just wanted to let you know there were some really good talks at FLEPC. So while we're thinking about scheduling speakers for next year, maybe midsummer we can start asking folks. I'll send you a few of the speakers' information from the conference. And we can talk about making up the new schedule for next year, because it would be nice to have that in place by early fall.
Yeah, absolutely.
And yeah, and if anybody has any suggestions, of course, we'll continue to ask for those as well. But yeah, if nobody has any questions or anything, it's fine to get off early too. But thanks so much, Emily. You did a fantastic job, and Susan for presenting. That was great. And Crystal, you guys rock.
I'm still here.
You do.
Thanks, Sherry. I appreciate it. We're working on it.
Yeah, no, you're doing fantastic things. It's
Now,
really
if we can
exciting.
just figure out how to feed the hydrilla to the snakes, the pythons, that would be nice.
Vegetarian pythons.
Yeah. Yeah. Yeah, for sure.
Yeah. The perfect storm.
Well, great. Thank you so much for your time today, everybody. And we will talk to you again next month. Have a great rest of your day.
Thanks, Emily. Fantastic job.
Good job.
Thank you.
Bye-bye.
Bye.